Okay, so let's breeze through muscle tissue, see what we can talk about here. And of course, this is all about skeletal muscle, right? This is all skeletal muscle. But for this exam, you do have to know the differences. So revisit that. And also, when you do the lab for muscle, which is starting next week, you have to know what they look like with histology too. I mean, you probably already did that on the first exam, but just know we're dealing with skeletal muscle, which is voluntary. It's striated. It's multinucleated, and the nuclei are on the peripheral. And I'll take a look at it if it's on this PowerPoint, but usually we've done this already by now. Cardiac muscle, I had this behind me before, and this is the one with the intercalated discs and the branching. Of course, it's involuntary. But both skeletal and cardiac muscle are both striated, where smooth muscle is not striated. And of course, it's involuntary. Smooth muscle is kind of spindle-shaped. And smooth muscle lines are, like you told me on the last exam, it helps pump blood out, through the vessels, not pump blood, but move blood through the vessels and helps us digest and all that. You did very well on that. Cardiac muscle, of course, is the heart muscle for contraction. Right? And the muscle cell could also be called a muscle fiber. There's nothing like a muscle cell that's completely different than any muscle cell you've ever seen. So go through these, know the differences, the basic differences, the striations versus non-striated, and smooth versus cardiac versus skeletal. Because most of our exam is going to be about skeletal muscle. All right, so here's the histology, very easy. No tricks, straight up. Cardiac is only the heart muscle. Cardiac muscle is branched with intercalated discs. Probably put that on a exam. So we went through this so extensively, talking about the excitability, resting membrane potential, depolarization, repolarization. So please revisit those recordings. And I'm going to go over it a little bit tonight as well. It can be stretched. It has elasticity, so it can comply and recoil back to its resting position, right? And know the basic functions. It generates heat, of course, but it stabilizes those joints you learned and maintains posture and, of course, movement. Smooth muscle is about digestion. Pumping blood is about the cardiac muscle. That I kind of talked about what a tendon is a bit. A tendon attaches muscle to bone, and you knew that was dense, regular connective tissue in the first exam. Ligaments connect muscle to muscle. I'm sorry, muscle to bone. I'm sorry, bone to bone. Ligaments connect bone to bone. I was getting ahead thinking about fascia, or aponeurosis, is connecting muscle to muscle. And we'll go through that in lab in a couple of weeks, right? Or next week actually. The coverings, I don't spend a lot of time asking you questions about this. So I kind of know EPIs on the outside. You have practice questions with pictures. Could help you with the lab, but you have so much other things to worry about for this lecture exam that I'm not going to spend a lot of time on this because we have to get to the sarcomere, right? So all this we're going to revisit in a lab. This is really important for lab 2, but I need you to know it for the lecture exam 2, what the insertion versus origin. So the insertion is the movable part of the joint. Like when you flex your biceps brachii, you're moving your radius. So the origin is the non-movable part, like your coracoid process of your scapula. Really important. Aponeurosis is a sheet-like covering, like the white you see in the muscle mat between the muscles. And a tendon, of course, is connecting the muscle to the bone. Usually the origin and the insertion, right? So just take a look at this before you go into the exam, do the practice question. So I might ask one question, but I'm not going to, live here. This is just talking the difference between a muscle fascicle and muscle fiber. We're going to live here a lot, right? Look, you have the sarcolemma now. You have the myofibrils, which have inside the myofibrils are the myofilaments, which is actin and myosin. Yeah. So outside the sarcolemma is the endomysin. Okay, so remember the terms. This all has to do with the sarco is muscle, means muscle. Lemma is the covering. or plasma membrane. It is a plasma membrane. It's biphospholipid plasma membrane with receptors and the same setup as the cells you learned. Sarcoplasm is the cytoplasm of the muscle cell. All right? Glycosomes are glycogens for glycogen storage, so you should know that glycogen is stored in the skeletal muscle, and myoglobin is storing oxygen. And of course, the sarcoplasmic reticulum. The thing you need to know most is that the sarcoplasmic reticulum stores and releases calcium, because you need calcium, and we'll see why again. Again, you listen to the recordings, you'll see it over and over, and we'll go over it tonight. So in the myofibrils, this is where we're going to see the myofilaments, which is mostly actin and myosin. So this is big volume, a lot of, it's all protein, right? This is, we don't want to use this for energy. This is our structure, protein. So this is a nice close-up. And then we did the sarcomere, right? Good practice questions, I think, on that. So you could see the dark A bands. Remember, there's an A in dark, I bands, I in light. They're multi-nucleated with nucleuses on the peripheral. And then we went into the sarcomere, right? There's the sarcomere. Remember, the sarcomere is defined from Z disc to Z disc. It's resting state, right? And we have actin, or the thin, light, and myosin are the dark fibers, like the golf clubs, where actin is more like the chain of beads. And there it is. So this is a good little cartoon thing you could see in the resting state of a muscle. And we did mention titin as a protein as well, dystrophin, we'll get to that. And this is the myosin, the thick fibers with the tail, right? The tail and the head is what's going to bind to actin. And when myosin binds to actin, you have a cross bridge. So this is about actin, right? You don't have to know G-actin versus F-actin for our exam. Just know actin, right? And then the two regulatory proteins, troponin and tropomyosin. Remember, calcium binds to troponin. All right, so here we have it. Close-up of those myofilaments. And remember, the first thing, before you could even bind myosin to the exposed binding sites on actin, you have to energize the myosin head with hydrolysis of ATP. And this is the troponin-tropomyosin complex. Remember, tropomyosin covers the binding sites on this actin. This is actin now, the beads. So when calcium binds to troponin, that will displace the tropomyosin off of the binding sites for myosin on actin. Here's some other proteins. I like to mention titin. So it gives it some strength and helps recoil. It's those little stretchy things at the end of the thick filaments, the myosin. Dystrophin, right? Muscular dystrophy, right? We talked about that. I'm going to want to talk about it. It's too sad. But of course, that's a genetic sex-linked condition where dystrophin is not made right. So that linkage of those actin in the sarcolemma will fall apart, basically, and they'll be infiltrated by fat, causing Duchenne's muscular dystrophy. Nebulin, myomycin, and C proteins, they bind filaments and sarcomeres together. So these are all structural proteins, but the key proteins are actin, myosin, troponin, and tropomyosin. Right? No dystrophin because it relates to that clinical correlation to this, Duchenne's muscular dystrophy. So read about that. Going on, it shows a picture, very sad. Okay. We go on long, right? So the sarcoplasmic reticulum is all about calcium. So after a muscle contraction stops, or if you want a muscle contraction to stop, the calcium is pumped back into the sarcoplasmic reticulum until it's excited again. Action potential will cause the calcium to be released from the sarcoplasmic reticulum and bind to troponin. Don't worry so much about the triad, but they're basically networks of tubules stemming from the sarcoplasmic reticulum, delivering ions to where they need to go, to around the sarcolemma to the next sarcomere and the next sarcomere. So it's a communication of ions, all right, especially with calcium. So this shows that network, pretty cool. You can see it's all part of the SR, all that bluish you see there. So this big thick piece right here is a Z disc right between those. And it's also a communication, actually. It's just thicker, slightly different connective tissue. Right, so this is a resting muscle. Know all the parts of the sarcomere. Right, H zone, M line, Z disc, I band, A band. Okay. So we'll start talking about muscle contraction. Sorry for the delay. I'll do a slideshow just to get us rocking. So here's some things I want to go over with you, like how a muscle contraction starts. Remember the action potential in the cerebral cortex. That's the brain, right? The outer part of your cerebrum. And when we do neuro, we'll talk more about that. And then you have an action potential in the somatic, I like the way this is showing up on the blue, somatic motor neuron. So this is a nerve cell coming from the brain. And the somatic motor neuron has an axon, right? That's been depolarized and went through action potential. And then you have the, let's call it axon terminal. So this is how it starts. You have a thought, right? A thought that causes an action potential in the somatic motor neuron. The axon of the somatic motor neuron conducts that action potential all the way to the axon terminal. The axon terminal has those synaptic end bulbs, right? The same thing, really, but they have vesicles inside, right, that contain acetylcholine, the neurotransmitter. So calcium channels open on the axon, voltage-gated calcium channels open. So calcium in where? In where? It's going into the axon terminal, the synaptic end bulbs. And then you could have vesicular release of the neurotransmitter, which is acetylcholine. So acetylcholine doesn't enter the cell. It diffuses to the other side of the synaptic cleft, and ACH will bind with its receptors.
What's ACH again?
Acetylcholine, that's the neurotransmitter. So you have all this electrical stuff going on at first, the depolarization. Then you have action potential, and that is carried from segment to segment to the axon terminal. So once the action potential reaches the axon terminal, which is the same thing as a synaptic end bulb, calcium comes into the synaptic end bulb. This is all electric voltage. Once calcium comes in, it can allow for the release of acetylcholine, which is stored in vesicles in the synaptic terminals. So acetylcholine is a neurotransmitter that's going to go from the nerve cell, in this case, to the muscle cell. So acetylcholine needs receptors, and the receptors are on the sarcolemma, the postsynaptic membrane. So once acetylcholine binds to its receptor, then sodium channels open and sodium enters the muscle cell. or the muscle fiber, whatever you want to call it, inside the membrane now. So that's depolarization. All right. So once that happens, the sarcoplasmic reticulum, because, well, actually, before that even happens, you have complete depolarization at the threshold, and you have a muscle action potential. And then the SR, sarcoplasmic reticulum, will release calcium inside the cell. So what does calcium do? Calcium binds to troponin. And that displaces tropomyosin off of binding sites on actin, binding sites for myosin, right? So now we're ready to go. Now we're ready to go. We're ready to start the muscle contraction, because now the tropomyosin is not blocking the binding sites on actin. So now we can do the steps of the sliding filament. So the first thing you need is your hydrolysis of ATP on myosin head. So that energizes the myosin head to configure it to get ready to bind to actin. So the second step in the sliding filament is the cross bridge formation. Between actin and myosin. So they're bound now, right? So once they're bound, then they're going to take that free energy and do what's called a power stroke. And the power stroke is going to fling the actin on both sides of the M line, both sides of the sarcomere, towards the middle of the sarcomere, towards the M line. So a fully contracted muscle, you lose the whole H zone. So the Z disc comes closer to Z disc on the other side because of the actin moving towards the middle of the sarcomere on both sides. And that's shortening the sarcomeres along the muscle, and that's muscle contraction. And we can't stay contracted, so the last step is the detachment. And for that, you need more ATP synthesis. So remember we said rigor mortis is where you don't have oxygen, so you can't make ATP. And if you don't have the ATP, you can't detach the muscle. So that's one way the muscle stops contracting. The second is the destruction of acetylcholine in the synapse by the enzyme called acetylcholinesterase. So remember what acetylcholinesterase. And you'll see it written out. I'm just shorthanding it right now. And then the calcium that was released from the SR goes back into the SR. And now the muscle can stop contracting. Okay? So go through the recording from last time too, because that talks about muscle twitch versus summation versus fused tetanus and unfused tetanus. Okay? So again, this whole thing is about contraction. Remember, the sliding filament model starts with those 4 steps: hydrolysis, cross bridge, power stroke, and detachment. Right? So it's all part of that. So your book does it slightly different. But of course, I'm asking you questions mostly from the book and from what I'm telling you in these recordings. So what happens during the contraction? Z discs are pulled towards the middle. The I bands get smaller. Z discs, of course, get closer, and the H zone disappears. And that's contraction. Right? So you know all the steps now. So this is a fully relaxed muscle. You could see some of that H zone in there. You see where the Z discs are, and then this is fully contracted. So they kind of came together. Relaxed, contracted, relaxed, contracted. So the myosin is pulling the actin towards the middle of the sarcomere. So the H zone's disappeared. Of course, you see more of the myosin because it's darker. The A band is dark. The I bands are light. H zone is more light in the relaxed muscle. You don't see it in the fully contracted muscle. So again, good question before about what is ACH? It's acetylcholine. That's the neurotransmitter that's released from the neuron. So this is a neuromuscular junction. There's a neuron going to synapse with a muscle membrane. And this is muscle contraction, somatic muscle contraction, voluntary muscle contraction, skeletal muscle contraction. So muscles need an excitation from the nervous system to contract. Nothing's going to happen without that depolarization, without that action potential. So we talked about the ion channels, right? So the ion channels, the sodium, potassium, and we did depolarization last time, so that's important to remember. That sodium in is depolarization. That leads to action potential. Potassium out is repolarization, which gets it back to resting membrane potential. And that's all due to these voltage-gated channels. But a chemical synapse involves ACH and its receptors. The receptors are across the synapse, synaptic cleft, on the muscle cells, specific for acetylcholine. So without the receptors, there would be no contraction, because acetylcholine would never bind. So this is a close-up of a chemical gated channel and then a voltage gated channel. So basically the receptor for acetylcholine is an ion channel, allowing sodium to come in and potassium to come out, technically. It's not a sodium-potassium ATPase pump, though. This is a ligand-gated channel, which you'll learn in neuro, but this is a receptor for acetylcholine on the muscle membrane. So the axon is the thread-like part that goes away from the cell body of the neuron. Right, so we didn't learn neurons yet, but hopefully, let me see if there's a picture on the other side. But this is ultimately the neuromuscular junction, where the nerve cell meets the muscle cell to make it contract via action potential and release of acetylcholine and binding of acetylcholine. So this is the axon coming down. It's all the same neuron, and these are the axon terminals coming in contact with the sarcolemma. So voluntary muscle starts at the cerebral cortex. And this goes into the axon terminals and what the synaptic cleft is. That's the gap junction between a nerve and a muscle in this case. But later on in neuro, you'll see they mostly between neurons. But for muscle contraction, you only need to know one neurotransmitter, and that's acetylcholine. And this is the neuromuscular junction, with the synapse is the space that the neurotransmitter diffuses through. Remember, the calcium channels have to open to get this neurotransmitter to be released. And that happens in nerve cells too. I mean, going to nerve cells. So sometimes they call this the presynaptic membrane, and this is the postsynaptic membrane. So presynaptic membrane. And then the postsynaptic membrane. So in this case, this is the neuromuscular junction. So the presynaptic membrane in muscle contraction is always a neuron. And this is the synaptic end bulb, or axon terminal, whatever you want to call it. These are mitochondria. These are all vesicles with neurotransmitters. So the postsynaptic membrane needs to have receptors, or else you're not going to get a change in the postsynaptic membrane. So here's the receptor for acetylcholine, which are basically sodium channels. So sodium can come in and excite the membrane, depolarize it to action potential, and that'll continue to the next and next muscle cell and all the sarcomeres that are in the muscle cells. So the sarcomere is the smallest part of the muscle. the biggest part of the muscle, of course, is the epimysium, the muscle belly itself, which you're going to start learning next week. All the different muscles. Just when you're starting to learn the bones, you have to learn muscles. Okay, so the big picture. Whoa. So again, don't forget, in order for this to get to the neuromuscular junction and skeletal muscle, you have to have a thought from your cerebral cortex. And that somatic motor neuron has to be excited and go through action potential. So excitation, contraction, coupling is basically happens at the neuromuscular junction to cause the 4 steps. This is 4 steps, remember? Hydrolysis, cross bridge, power stroke, and then detachment. So this kind of goes through that. Pretty good, starting at the brain, all right? So I like this one. I like this one because it gets you to the cross bridge, the connection. It talks about the calcium. The only thing it doesn't tell you here is about the calcium channel here opening. Because you need calcium to come in for muscle contraction. Really important, action potential. So here it is. Action potential arrives at the neuron's axon terminal, or synaptic end bulb. And here's the point I was trying to make. This is important. Look at this. This is where everybody misses. This is at the axon terminal. The voltage-gated calcium channels open so calcium can enter the synaptic end bulb. And that is what causes the release of ACH, which is acetylcholine, and it is a neurotransmitter, into the cleft. It diffuses to its receptor, which are basically sodium channels. And then acetylcholine binds to its receptor and opens those gates, and off it goes, right? Off it goes to muscle action potential. So acetylcholinesterase will stop the release of ACH, so the muscle contraction can stop. Really important. Another nice close-up of that, the events at the neuromuscular junction. And see what action potential causes. The voltage-gated channels open, so I really like this little slideshow here. Shows you everything. All what we just did. Then ultimately, that binding of that acetylcholine opens the sodium channel, so now the muscle membrane can depolarize to an action potential. Then you'll get muscle action potential. Not easy. So once you get the muscle action potential, then you're going to have that release of calcium from the sarcoplasmic reticulum. And once calcium is released from the sarcoplasmic reticulum in the muscle now, then you'll get the binding of calcium to troponin, and then troponin moving that tropomyosin off of the binding sites on actin for myosin. And then you could start the 4 steps. First step, hydrolysis of ATP on the myosin heads. Second step is the cross bridge between actin and myosin. And the third step is the power stroke, shortening the sarcomere, bringing the actin towards the middle. And the last step is detachment, which needs ATP. So to stop the muscle contraction, you have detachment, you have sequestration of the calcium back into the sarcoplasmic reticulum, and you have the enzyme, acetylcholinesterase. We'll revisit this. I'll start the nervous system next time. Okay, so let's go to the next one. I'm sorry, I paused the recording somehow. The next one is about action potential. And let's go through some of this. I did this last time when we started the other thing. So again, I'm not going to go over every slide, but I just want to give you some highlights. The next one is the next slideshow is about, remember, concentric and eccentric, isotonic, and then isometric contraction, and all the muscle twitch versus summation, fused tetanus, and unfused tetanus. So we did that last time. So that one, just listen to that recording when you get a chance again. So action potential, right? Depolarization and repolarization. So all happens of the membranes, right? With these now chemically gated ion channels need a neurotransmitter, right? And that's ACH. So the voltage gated is mostly about sodium and potassium. So let's see if we got a good picture coming up here. This is both, right? We've seen this, the axon terminal, calcium channels on the neuron, and the postsynaptic membrane. So here's depolarization. Remember, depolarization is sodium in, and the sodium gated channels have to be open to allow sodium to come in. So that all starts from a previous action potential. So now we get a new action potential of, say, a muscle cell, right? So depolarization is about large influx of sodium from a stimulus, right? And if it reaches threshold, you'll get an action potential. So this is different channels along the axon, right? When sodium channels are open, you get depolarization. So the inside can become more positive, which is happening here. And then if you reach the threshold, you get an action potential. Potassium stays in until it's ready to repolarize, because repolarization is potassium leaving the cell, right? So repolarization is about efflux. And we mentioned refractory is a period during this action potential winding down where another stimulus is not going to get another action potential. That's what a refractory is. So you have to know what the resting membrane potential is, and that's used, and that's maintained by those sodium potassium ATPase pumps. Same thing. Okay, so this is where you want to live. We went through this and go through that recording and see, like in a muscle, the resting membrane potential is minus 90 millivolts, right? Once the sodium influx, you get depolarization. Then it peaks out at an action potential. This is the action potential. So that's all or nothing, and that'll continue down the length of the axon. So it has to reach threshold, though, right? And the threshold, I think your book says minus 65 millivolts, minus 65, that is. So if you get enough sodium in to reach that minus 65, then it's all or nothing action potential. And it keeps depolarizing, of course, till it peaks, and then the potassium leaves the cell, and that's repolarization back to resting membrane potential. And then these channels close, and potassium stays inside. It doesn't leave anymore, unless it has to be hyperpolarized. But we'll talk about that in neuro, about hyperpolarization of the resting membrane potential. So excitation, contraction, coupling. Of course, you need the action potential to get to the muscle, or it's not going to do anything, right? And one of the main things is the calcium release from the sarcoplasmic reticulum, and that leads to contraction. You could probably tell me how now. In the muscle, it's binding to troponin. Right? So action potentials are brief, very lightning fast, all or nothing, like flushing a toilet. Okay? So go through that part from the last recording, and we'll continue with bone in a minute. Okay, let's go through bone. Just the highlights. Again, there might be stuff on here that I'm not saying now that I did in the other recording, so please go back and check it out. All right, so this is good too, because you're doing your lab practical after Sunday. So let's get into it. Okay, remember the skeletal cartilage, basically the costal cartilage, right? The pubic symphysis, the articular cartilage, synovial joint cartilage, right, and so on. So no difference between the cartilage and the bone. Not much elastic cartilage on the bone, but elastic cartilage is part of the epiglottis, and we already tested you on that. Fibrocartilage comes back, though, because of the joints and the pubic symphysis. So that's really important to know the difference in that. So a general look at the skeleton, and most of that is in lab, but these are the things, like appositional growth is growth in width, right, that can continue after the growth plates fuse. Interstitial growth is growth in length. That has to do with the growth plates. All right, some of the functions, remember, for support, protection, movement, mineral storage, phosphate and calcium, but especially hematopoiesis, right? Making blood cells, very important. And the yellow marrow stores the triglycerides and produces a hormone called osteocalcin, right? Helps to regulate the insulin secretion and the glucose levels. Axial versus appendicular, we really went over that very carefully in lab. So know the difference, even, and more for lab, like know the difference between long bones and short bones and some examples of each. So in that lab 6, I believe it is, has the information PowerPoint on all the classification, flat versus irregular. Remember the atlas and axis, know the difference between those two. And then the os coxa, kind of irregular, right? So gross and microscopic. You kind of did the microscopic in histology in chapter 4, so that you might revisit that on exam 2A, but you already did it in lab. So this is definitely more physiology of bone that we're talking about for this exam 2B. Compact versus spongy bone. Please know all the little details with that. All right, and where you see it more, like spongy bone more in the epiphysis, compact bone along the collar or the cortex of bone, especially along the diaphysis. So of course, know the parts, the diaphysis and epiphysis, proximal and distal, and the long bones, especially. What periosteum is and endosteum. Trabecula is more for spongy bone, right, and diplo. So it's more trabeculated. This is not osteoporosis. This is normal. It looks kind of porous, but this is spongy bone. Know the magnification, right? This is really normal looking here. Scared me for a minute. Know the difference between epiphysis and diaphysis. And that epiphyseal line is when the growth plate fuses. So you know that's an adult. And most of the bones we see are adult bones with a line. Epiphyseal plate is the growth plate, which is cartilage. Again, showing the parts of the bone. Just make sure you know the basic parts. Go through those study questions, practice questions. Know that articular cartilage lines the ends of the bone, proximal and distal, to form the joints. This is a nice view of compact versus spongy bone. So good visual. You can see the arteries coming through. And know the membranes, periosteum, osteogenic. So we're going to get into more osteoblasts and osteoclasts and osteocytes. You really have to know those well. And the endosteum again. Delicate connective tissue. All right. More pictures. Red marrow. Okay, so red marrow is important to know that that's where the hematopoiesis takes place. So you see that, of course, in the medullary cavity, not just in newborns, but in everybody. Bone markings, all those crazy things we saw in lab, all those little parts we had to name, which are sites of attachment for muscles and via tendons and ligaments. So again, bone markings, this is more about lab, so please revisit all this. And this is really important here, the different cells. The osteogenic cells are basically the stem cells, which you already know they're going to be bone. Osteoblasts, remember, they're building bone, laying down collagen. Osteocytes are the resident cells, right? The functional cells, the mature cells. The bone lining cells are basically osteoblasts and osteoclasts and more stem cells. And then osteoclasts are the ones that remove the calcium. So that's like a white blood cell. Yeah. So osteoprogenitor and osteogenic are the same. They're basically the stem cells. Osteoblast laying down the matrix. Because remember, bone is connective tissue, right? Sparse cells with a matrix, mostly calcified matrix. So the osteoblasts are turning over a lot, highly mitotic. Some pictures of our friendly cells. Osteocytes are the mature cells, the one that hang out in that lacunae, right? In that extracellular fluid within the compact bone and spongy bone, of course. Osteoclasts are more destructive, but appropriately destructive unless they get out of control. Osteoblasts more building, right? Bob the Builder is osteoblast. Wreck-It Ralph is the osteoclast. Again, these periosteal cells and endosteal cells are basically osteoblasts and osteoclasts just working together and maintaining the shape of the bone and the density of the bone. Again, these are the osteoclasts, more resorbing bone. It's called resorption, and that means take the calcium out of the bone. Resorption, not reabsorption, resorption. More pictures to look at difference from a different lineage, from white blood cell lineage, osteoclast. This in histology, real histology, those cells. Compact bone, remember osteon is the unit of compact bone, the central canal or Haversian canal, the canaliculi, taking in the oxygenated blood and deoxygenated blood out. And they have circular or circumferential lamellae. So let's take a picture of that. This is like, I would never show you this picture on a lecture exam, but that's just showing you the osteon. And the central canal has all the vessels, the blood vessels, the arteries and veins, and some nerve tissue too, and lymph. The Haversian canal is the central canal. The perforating canals come through from the sides through the periosteum into the cavity. So bone is very vascular, a lot of blood supply. Know what lacunae and canaliculi are? Not so important. Circumferential is rounded, and interstitial is more in between the rounded ones. So not that important to differentiate those two. So this is all the lamellae, and you see the central canals. Each one of these circular units is an osteon, and this is compact bone. You probably saw this on the first lab practical. Okay, spongy bone is more about trabeculated bone, like that porous looking bone. It has more marrow in it, so the marrow is more in your spongy bone areas, just like we saw before. Again, same basic cells, right? And this is the difference between, the organic part and the calcified part. So bone is mostly the calcified part, and that's called hydroxyapatites, which they're inorganic. Like protein is organic, and that's what's making up the rest of the bone. So that's collagen, right? Because this is connective tissue. So hydroxyapatites are the mineral salts, the calcium carbonate, the calcium phosphate, right? The minerals, calcium, phosphate, fluoride, magnesium. So that's called hydroxyapatite. Right, so that's inorganic, the minerals. So let me go through the next one. Okay, so part 2 of the bone. Ossification, right? Osteogenesis. And I'm going to stick with endochondral ossification. I'm not going to do intramembranous much at all. Just know that endochondral is about long bones, right? Based on a hyaline cartilage model. Intramembranous is more about the flat bones and irregular bones. So the long bones, are like the phalanges and the humerus and the femur and the tibia, all those that have diaphysis with proximal and distal epiphysis. So this is the one I need you to learn, more so than intramembranous, but just know the difference. Okay, so this is about the long bones, starting with the hyaline cartilage model and the primary ossification center and the 2 secondary ossification centers. So we get a picture of that, and, where it starts out as the hyaline cartilage model as an embryo, right? This is before you're born. And there's a primary ossification centers right in the middle of the diaphysis. So it all is hyaline cartilage before it even calcifies. So ossification is the calcification part. And then this starts to get bigger, and the osteoclasts help out, and the chondrocytes, or cartilage cells, they begin to die. And this gets taken over by osteoblasts, building more bone, and then osteocytes ultimately as it calcifies. And then you get some more blood infiltrating, so you get more calcium into there and oxygen, so the osteocytes can do their maintaining. So you see like it's growing in length, and that's interstitial growth. Still ossifying though. And then you have two secondary ossification centers in the proximal and distal epiphysis as it keeps receiving blood. And then ultimately this all calcifies, and you're left with these remnants of cartilage between the diaphysis and the epiphysis. And that's your epiphyseal plate, also known as the growth plate. And that continues to grow until, if you're a female, probably till you're about 19 or so. And then in males, a little bit later, 20, 21, it just varies though. So that's your growth plate, which is cartilage. And you can see you're left with a remnant of articular cartilage, which is also hyaline cartilage. So now the calcification is basically done. Now it's just about interstitial growth. You know, of course, this is going to fuse and stop growing when you, after puberty, right, into adolescence. So this basically stops proliferating cartilage after very high spurts of testosterone in the male or estrogen in the female. So intramembranous ossification, we're not going to talk about much, but that starts directly from mesenchymal cells, which are connective tissue stem cells, whereas endochondral ossification is more about the hyaline cartilage model. So endochondral ossification is for your long bones. Intramembranous ossification is more for your flat bones, like the parietal, frontal, temporal, and irregular bones. So clavicle is not a long bone, just so you know. Okay, same kind of the way it's laid down. You're going to have osteoblasts, you're going to have osteocytes, but just a different configuration by the way that bone ossifies. So know the endochondral ossification well. Okay, now appositional growth, again, is growth in thickness. Interstitial is growth in length, and that has to do with the epiphyseal plate. Very important. So this is talking about the different zones within the growth plate. And you should know those. There's probably questions about it in the practice questions about the different zones, the resting proliferation, hypertrophic, and calcification zone. Okay, and this is the picture of it. You can see these are growth plates here in a young knee. Like this is the tibia. Can't see the patella, probably because the x-ray went right through it. This is an x-ray. I'm not going to put x-rays on the lab practical. It would be rare, but I mean, it's possible, but they'd be very, very straightforward. So here's your zones. Again, no, that's the growth plate. Yeah, so this, again, this is just general. You don't have to know exact, but close. Know that the males, the fusion and epiphyseal line shows up later than females. Appositional growth, thickness, right? More pictures, which is helpful. So the hormones, now this is important. So growth hormone technically is the most important for growth of the epiphyseal plate. Thyroid hormone is also involved with anabolic building of bone as well. Testosterone and estrogen, very anabolic, means they increase the building of bone through protein synthesis. Right? And this is when these spurts go on, that's when the closure of the epiphyseal plate happens. And the epiphyseal plate, again, is the growth plate. Okay, bone remodeling. But I think we went through fracture repair too. So osteoclasts, again, they break down the bone, basically taking the calcium out by dissolving it with hydrochloric acid, really, which is part of an enzyme. So here's the PTH. So PTH is released from the parathyroid gland, which is behind it's embedded in the posterior thyroid gland. So this is parathyroid gland. PTH is the hormone, and that activates osteoclastic activity. All right. So this is very important. When PTH is released, the osteoclast will increase the taking of calcium out of bone and putting it into the blood. So PTH, when released, increases your blood calcium. That's very important. PTH increases blood calcium. So let's see another control of hormones. Again, this is just going on about the same thing. Now, calcitonin is released by the thyroid. gland. And calcitonin responds to high calcium levels and blood calcium levels, and calcitonin lowers blood calcium levels. So calcitonin puts the bone calcium back in the bone. So they're kind of like completely opposite. But remember what gland they are. This is not the thyroid gland. This is the parathyroid gland. Calcitonin is released from the thyroid gland. So two different glands. Parathyroid hormone responds to low blood calcium levels by increasing blood calcium levels by stimulating osteoclasts. This is very important, all this. Calcitonin responds to high blood calcium levels and decreases the blood calcium levels by depositing the calcium back into bone. So parathyroid hormone utilizes osteoclasts, Wreck-It Ralph. Calcitonin utilizes osteoblasts, Bob the Builder. Okay, so that, of course, you want to get a balance in your homeostatic blood levels of calcium, because you just learned about how important calcium is in muscle contraction. And you're getting a good start on why it's important in nerve conduction as well, based on that synapse, the synaptic end bulb and the sarcoplasmic reticulum. Yes, right, so this is really important. So your body doesn't care how dense your bones are with calcium. They're more concerned about homeostatic blood calcium levels for things like muscle contraction and nerve conduction, and other things too, like blood clotting, fertilization. Again, so this is like hypocalcemia. Whenever you see emia at the end of a word, that means it's in the blood. Like hypo means low, calci means calcium. So hypocalcemia is low blood calcium. Hypercalcemia, hyper, too much calcium in the blood. All right? So it has to be homeostatic or it's not going to work correctly. Hypocalcemia means that it's going to be hyper-excitability of the neuromuscular junction, like too jumpy or can't be controlled. Hypercalcemia is going to just kind of stop everything because it's out of homeostatic range. So it's really important. And excess calcium, hypercalcemia, can lead to kidney stones. And calcium salts build up in the kidneys because the kidney filters out all of your blood, including the glucose and the calcium, phosphate, sodium, potassium, and other waste products. So it can affect the kidney and cause stones, caliculi. It's called renal caliculi is another name for kidney stones, right? Not the Rolling Stones like Keith Richards, but the kidney stones. Okay, so the hormone's very important. Here's a couple other hormones that are fun to talk about. Leptin is a hormone released from adipose. Get that, from your fat cells, right? So it may play a role in bone density and inhibiting osteoblasts, right? But I'm not going to talk about that. I'm not going to really ask you about leptin or serotonin on this exam. But when we get to neuro, I'll mention these again. Leptin technically is a hormone, but serotonin is a neurotransmitter. But we can talk about how leptin affects the brain. All right. So again, these two do have something to do with calcium homeostasis. But it's kind of when I see may play a role, I'm not going to really teach that yet. Right. But serotonin is released. I remember mentioning this to you. And it's made in the gut too. The serotonin's all over the place. So again, it may block bone calcium, and new calcium is flooding the bloodstream. When new calcium is flooding the bloodstream. So kind of that helps maintain the homeostatic calcium. Wolff's Law. I like Wolff's Law because Wolff's Law states that if you put stress on a bone, right, like exercise, weight-bearing exercise, then the bones will correctly build and take up calcium better, more osteoblastic activity. Where if you're sedentary, you sit down all the time, or if you're in space where there's no gravity, then your bones will get weak and maybe osteopenic or osteoporotic, which means less bone density. Yeah. Bending stress, I'm not going to ask much about tension lines in bone. Okay. Fractures. Now we kind of have to know the steps of the fractures, right? And go through these: displaced versus just non-displaced, complete, incomplete, open, and closed. And then there's different types of fracture, like comminuted. You see it broken into pieces, right? That's comminuted. Open fracture means it went through the skin, right? But this is comminuted, not necessarily open. This is a compression fracture, like a vertebrae. So this person looks like they have osteoporosis, so it makes the bones weaker. So just sneezing can cause a compressed vertebrae like this. It's called a bone compression fracture of the vertebrae. It looks like a little fish, and they're supposed to be nice and boxed looking, like you saw in lab. This one I'll ask you. This is a spiral fracture, mostly from twisting during sports activity. It could be closed fracture. Epiphyseal plates, right, where the actual bone diaphysis kind of shifts off of the epiphysis, and Emily could tell you about this, where it's a Salter-Harris fracture, but epiphyseal plate fracture, and you're immobilized for a while after this. Depressed fracture, not so important. This one I'll ask you though, green stick fracture. So this is in a young person. Obviously, you see the growth plates. Not so obvious, I won't ask you that, but so when the bone kind of splits like a green stick, and it kind of splits a little bit, but you could bend it a little bit back, and it kind of takes its shape again. So it's like a much more flexible bone in a young person that has this type of break in the cortex. That's called a green stick fracture. So of course, you had to treat it with immobilization, or it could be surgery sometimes with pins and wires, right? So here's the steps you need to know, the 4 steps of fracture repair. Remember right away, the first step is hematoma. Like when you're taking the test you're doing now, the tissue test, the first step in healing is inflammation in tissue. But the first step in fracture repair is hematoma. And then, of course, cartilage is going to come next because cartilage always comes before bone. And then you're going to get bone, and then the osteoclast will do the bone remodeling. It's kind of a memorization, but it kind of makes sense. First, after you get a fracture, the blood comes in to heal and protect and cause an immune response. And then cartilage will infiltrate to start to build the bone. Then it'll calcify, forming bony callus, just like ossification. And then it'll be remodeled by the osteoclasts. So hematoma, which is red because the blood vessels get more permeable, and more chemicals come in to help keep it clean and help repair it. So hematoma is the first, and this is what hematoma looks like after a long bone fracture here. A finger, maybe, a metacarpal, possibly. I'm not saying it is, but it could be. All right, then you have your fibrocartilaginous callus, where fibroblasts secrete collagen. That's what's happening. They're building that really cartilage first. And it's fibrocartilaginous, so it's not very good tissue. It's kind of cheap material that comes in first. So it builds that cartilage. So it's not bone yet. It hasn't ossified yet. So it's early on in the stage of healing a fracture. So this is the second stage. And it becomes hard. Now you get the bony callus. This could take about 2 months, right? So you have calcification now. So it starts to look like bone a little bit, but it's kind of all over the place. So that's the third step, is bony callus. The last step is remodeling, right? So it's got to start to look a little bit more. So it kind of, and you really need osteoclasts to clean that up. It's not telling you that, but they have to kind of clean up that excess bone. And of course, it doesn't happen. You're going to be left with scar tissue unless this is set completely right. Right? Scar tissue is like that too much fibrocartilaginous callus, too much extra bony growth.
Professor,
Yes.
I have a question. So, those arteries in the bone, can they get like damaged if you like fracture or break the bone?
Yeah, they can. You're right. They can. And these are very small vessels, so they do have a way of repairing themselves. So there's going to be excess bleeding, but they clot very quickly, and they become more permeable to like white blood cells can get out of there and calcium can get out of there. So these are really smaller than they look. They're more like capillaries, which you'll learn. So yeah, that's what really causes the hematoma, like you said. It's first of all, it's the damage of the blood vessel, and then the blood vessels become more permeable. That means like the blood vessel is going to release like white blood cells, going to release histamine and macrophages, all things that are supposed to clean up the cell and send the signal out. So yeah, that's very true. They get damaged, but they heal a lot faster than the bone. Believe it or not, the blood vessels in that these small, especially in something like a metacarpal. Good question. So here they are, all the 4 stages. Know them because we have a couple of questions on that. Some diseases, right? Osteomalacia and rickets. Rickets is mostly in children with very low vitamin D. And of course, remember, vitamin D helps with the absorption of calcium in the intestine. Osteomalacia is a bone softening disease more in adults. Osteoporosis is a bone weakening and loss of calcium, decrease in density. Paget's disease is basically too much calcification that kind of makes the bones very hard to move and very stiff. Let's see if we have a picture. Again, remember vitamin D. And in the exam 2A, the tissue and the skin, we talk about vitamin D being converted in the skin. Not vitamin C, Anthony. Vitamin D. Okay. So rickets, osteomalacia, osteoporosis. So this is osteoporosis. See, like the trabeculae are kind of the same size, but look at the in-between, like the actual bony part is kind of falling apart, getting weak, and that's osteopenia or osteoporosis. Risk factors is interesting. Just, decrease in estrogen really is one of the causes, because estrogen is very anabolic to build bone and maintain bone. So women, of course, are more affected, and when they get older, postmenopausal after they stop ovulating and menstruating. So it depends on the age. That's very varied, which you'll learn in reproduction. Other risk factors, always smoking. I think smoking is a risk factor for everything, as is diabetes mellitus. You're going to see that. So go through these things because it's kind of interesting. Clinical correlations with bone. Treatments. And this is one of the most important one early on is weight-bearing exercise, because that's Wolff's Law, right? That'll help build the bone, osteoblastic activity. So hopefully you have enough calcium in your diet. You don't have to supplement it. But vitamin D, people seem to be very low on vitamin D. Maybe we don't get enough sun. So this has to be replaced a lot and supplemented. Hormonal therapy is very dangerous if you supplement estrogen, because that will definitely lead to breast cancer with extended use. Not definitely, but it increases your risk of breast cancer. And if it's converted to testosterone, it can increase your risk of heart attack and stroke. These are the drugs. You don't have to remember these. These are kind of common. So this one, but look what it does. This is important. Look what it does. It decreases the osteoclastic activity, because remember, osteoclasts will resorb the bone, take the calcium out. And remember, PTH causes this. So too much PTH can cause osteoporosis, excess. This one is shown to reduce fractures in men with prostate cancer. But again, this works more on the hormonal part. You don't have to know the names of denosumab. Interesting. Reduce carbonated beverages, right? Because that kind of causes calcium to bind with the carbon and the carbon dioxide, really, which is, you don't really have to know that, but it's kind of an interesting point. And this is important. Plenty of weight-bearing exercises. I think that's one of the most important things for prevention. So Paget's disease is excess bone deposition, too much calcification, right? And the cause we don't even know, right? Viral possibly. So it's kind of rare, but you see it. So calcitonin, again, will lower the blood calcium. So it doesn't make that much sense because you're thinking, well, the calcium's going into the bone, but it still lowers the blood calcium. So there's less having to come into the bone. And also the one that you gave to the osteoporosis person. So this is what it looks like on x-ray. Like this is a normal x-ray. It's just extra whitening. So on x-ray, things that are denser look whiter, just in case you're wondering. And then it kind of falls apart. So really this is the problem. Not so much the darkening or the density, it's more the excess weakening in other places. So it's a total misdistribution of calcium throughout the bones. And I don't know why this is caused. It's always in an x-ray form. It's an x-ray phenomenon. Really. Development of bone. Again, you don't have to know too much about this. Just the endochondral ossification. Okay.